Thanks so much, Pablo, for the introduction. Thanks for the invitation. It's very fun to be here. I just wanted to give an introduction to the activities of my lab. If you don't know our work, we are broadly interested in regulation of gene expression in mammalian cells. And we use methods, statistics and computation, but especially machine learning, supervised to try to understand mechanistic information about gene regulation. And I'm actually only going to talk about transcriptional and epigenomic programs today in the context of dysfunctional tumors. But I just wanted to do a lot of work also on post-transcriptional regulation, co-transcription, alternative cleavage and polyadenylation, microRNA-mediated regulation. We also do some work trying to understand tumor data of big cancer immunogenic neoantigen. And you adoptively transfer CD8 T cells specific to this neoantigen into the mouse. All right, so you turn on the tumor, you transfer T cells of the right specificity, and you watch as the T cells turn to dysfunction. And all these mice will eventually get full-blown liver cancer, right? So even though the T cells are there, have the right specificity, they become dysfunctional. They don't stop tumor growth, okay? And she characterized functionally that there are two discrete states, okay? So early on, you have a plastic state. So the T cells are dysfunctional, but you can still rescue them with immunotherapeutic strategies. Okay, so for example, PD-1, or you can expose them to IL-15, a cytokine, and you can rescue their function. All right? But a little bit later, dysfunctional state, they can't be rescued anymore. Okay, so the same trick you used to rescue them earlier doesn't work anymore. All right, and just a little bit more of that data. All right, so here's the mouse model. You induce the driver oncogene. In 8 days, you can see by flow that the T cells are already marked. Treat them with IL-15, and they will secrete effector cytokines again, and they'll have cytolytic activity, and they will, work. Okay, so they can have a functional response, they can be rescued. But after about 30 days, these same T cells still look dysfunctional, still no secretion of TNF-alpha or interferon gamma, but they can't be rescued anymore. Okay? And the idea is that 30 days in, very early, so there are no clinically apparent lesions at 30 days. All right, so very early, you're triggering some kind of program, a differentiation program, where the T cells get to fix dysfunction, don't work, and don't do anything to kill the cancer cells. Okay, so where we came in, okay, and this is the project I'm going to spend the rest of my time on. We signed up to help Andreas Schietinger explore the epigenetics that might encode these different dysfunctional states. All right, so what we did was we did ATAC-seq profiling, so mapping Chromatinaccessibility in the liver cancer model at different days, different time points, and also in normal T cell differentiation, where you have the same T cells, the same antigen, but they're presented in a bacterial infection model. So you engineer Listeria to present the antigen, and you get normal effector and normal memory T cells. All right? And we also had RNA-seq in all these cell types. All right? And I'm not going into many details, but we spent a lot of time figuring out how to rigorously analyze chromatin accessibility data. One of the first steps is we create an atlas of peaks that are accessible peaks, chromatin-accessible peaks or events that are reproducible between biological replicates in at least one cell type or time point. And when we do that in this data set, we get an atlas of about 74,500 peaks, okay? Okay, and this just came out actually a few weeks ago. All right, so the big picture of this analysis is here. If I represent every experiment, every cell type and replicate by its accessibility pattern, accessibility profile across the atlas, and I do PCA, I see that the normal cells are here, naive effector memory. So those are all in green. In the tumor model, in the liver cancer model, there's a big first step, day 5, day 7, where already I'm in a dysfunctional state. I'm not sort of in a normal day 5, day 7 state. And then there's a second big step between day 7 and day 14 from which you never return, okay? All right, so it seems like we have these two discrete states. They coincide in time with the functional states, plastic and fixed dysfunction, but we need to explore that further. Just if you are not familiar with these kind of data sets, this is quite typical that only about 20 to 30% of the peaks, so in this case 22% of the peaks, are at promoters. The rest are about half and half intragenic and intronic regions, okay, and just a little bit of peaks over exons. But if you look at peaks that have significant changes in accessibility between cell types, then the action tends to be at the intragenic and intronic peaks and not at promoters, okay, with some important exceptions. Okay, here's another view of the same data. Here, what we're doing is we're looking at chromatin-accessible loci in the atlas, or atlas events, and every row is a locus in the genome. Red is open, blue is closed, and we're seeing, as we go through this time course, what the accessibility changes look like, okay? And you see there are two states also from this picture, okay? So just a different way of presenting the same data, this early state and this late state, day 14 and on. And the big changes are naive to day 5 and day 7 to day 14. Okay, that's shown here. Okay. All right. Just to show you that this data is very beautiful, right? And you can really see with great granularity potential regulatory elements. Okay, I'm showing you two loci, the locus encoding interferon gamma and the one encoding PD-1, okay, which is the target of 23 anti-PD-1 drugs, all right? And you can see around this locus, these accessible events, normal, differentiation, naive effector memory, and then the time course in the tumor. And you see that there are events that are both on-off and quantitative differences between normal and dysfunctional differentiation. Okay, so for example, here is an event that is closed and naive, opened up in effector cells, maintained in memory normally, but it's never established as an accessible site in the tumor model, okay, in the CD8 T cells in tumors. Here's a site that is small and naive, grows very accessible in effector and memory cells, and it starts out as accessible, becomes too small, so there's a quantitative difference for that accessible site. Okay. Here, PD-1, again, there's an event here that seems specific to the tumor, an enhancer, perhaps, that you only see in the tumor. Here's an event that should be big and is small. All right? Okay, so I want to just show a little bit more of the data, when you have these data sets with Chromatinaccessibility and also gene expression profiled by RNA-seq, everyone will want you to correlate the two, and they'll use that word. Can you correlate accessibility and gene expression? Okay, and that's actually, it's a complicated question, partly because it's not a one-to-one thing, right? So a gene locus has many peaks, and you can do different kinds of statistics, or you can do supervised modeling, supervised learning modeling, or you can show a picture. So I'm going to just show you a picture, and I'll see if this makes sense. Okay, so here I'm showing both accessibility changes and gene expression changes of the top 50 genes that go up or go down in this transition from what we think is plastic dysfunction, day 7, to fixed dysfunction, day 14, okay? At least it's this big chromatin accessibility change, a change in chromatin accessibility states. All right, and I'm just ordering them along the x-axis. The y-axis, y-axis tells me the fold change, okay, the log fold change. So these are, I guess I don't have the, I don't have the scale, but these are pretty robust fold changes. But above the point, I have a stack of diamonds, and every diamond represents a chromatin accessible site. Orange means the site is becoming more accessible. Blue means the site is becoming less accessible. Okay, so visually, what you can see is that in this state change, okay, day 7 to day 14, you have a bunch of genes that are going up. They tend to have a bunch of peaks that are getting more accessible. The genes that are losing expression, their locus seems to be closing down. They're losing accessibility of many peaks. But there are also peaks that do the opposite thing. It's not perfect. Yeah? Some of these genes whose locus is closing down or opening up and that are having pretty big expression changes are themselves transcription factors, okay, affecting regulation of other genes, including BLIMP1 is a famous transcription factor, and LUF1, which is a TCF family transcription factor that we'll talk about in a minute. Okay, so what can we do with chromatin accessibility data that we couldn't do with RNA-seq? Okay, so certainly we can do a PCA plot with RNA-seq, but if you have the accessible loci, you can do a statistical analysis of DNA binding signals for transcription factors. So you can start to get at differences, potential differences in transcriptional regulation, what's binding, what's occupying the chromatin between normal differentiation versus dysfunctional. Okay, there are different ways of doing this. What we did for this paper, we took all the transcription factors that were expressed in our data set. That's about 200. For each one, we assigned a unique DNA binding motif, okay? And that's a whole procedure in itself. So some transcription factors will have 10 different motifs in a database, and you have to impose a hierarchy of believability, okay? Once, for example, you associate a transcription factor like NFAT-C1 to a motif, you can use that motif to index into your atlas, find all the peaks that have the motif. And then you can look at the PDF, right? Sort of a histogram of accessibility of those peaks, right? So what we're doing is something like for every peak that has the motif, we're looking at the height of the peak in a cell type and looking at the distribution of those heights over all the peaks. Okay, so different colors are different cell types. These distributions are bimodal. Basically, oops, I'm sorry, this is a little tricky. Basically, you have peaks that are closed and peaks that are open, okay? And what you can see is that the NFAT-C1 occupancy gets large early in the tumor, if I can do this without advancing, okay? And that doesn't happen normally, all right? So there's a large number of peaks carrying the NFAT-C1 motif that are opening up early in the tumor. And you can do a statistical test to say, what are the transcription factors that are, their peaks are opening in day 5 in the tumor versus day 5 in normal differentiation, okay? And in fact, you get NFAT-C1 and a whole bunch of other NFAT family transcription factors, okay? And there are other things that, many things are happening. There are other transcription factors whose program is closing, but I'm just gonna focus on this and have you remember the NFAT family transcription factors opening up. Later, as we go from what we think coincides in time with plastic dysfunction to fixed dysfunction, day 7 to day 14, other things are happening, okay? So for example, LEF1, this transcription factor I told you about before, if you look in the tumor, you see that late in the tumor, there's a collapse of the LEF1 program, okay? So many peaks that have the LEF1 motif are getting inaccessible, okay? So that's the signal you see here. And in fact, if you look at what are the changes in that transition from early to late, day 7 to day 14, it's LEF1 and also a bunch of TCF family transcription factors that seem to be losing their accessibility. You're losing that program, okay? Okay, so we wanted to try to validate some of these predictions. So from the ATAC-seq analysis, we get these transcription factors. We think that, well, statistically, they're associated with accessibility changes, early tumor versus early normal, and late tumor versus early tumor. How can we show that they matter in driving this differentiation program? So what we did, or what our collaborators in the Schiechinger lab did, was they used drugs to modulate the activity of transcription factors. So broadly speaking, what's going wrong is we have too much activity of NFAT family transcription factors early, and then later we have a loss of activity of TCF family transcription factors. So what they did was they tried 2 drugs. So they first tried this calcineurin inhibitor that inhibits nuclear translocation of NFAT, so reduces the activity of NFAT. And their treatment was every day up to day 8. And then they also tried that together with a second drug that activates Wnt beta-catenin signaling to increase TCF1 activity, okay? And they tried that a little bit later, up to day 8. And then day 10, they take the T cells out and try to do some functional evaluation. All right, so what you can see is that if you look at this day, day 10, the PD-1 levels in control versus the drug treatment, you're getting less expression of PD-1, so they seem less dysfunctional. The T cells are less dysfunctional after the drug treatment. They're expressing more TCF1 and more EOMES, so these are both important effector transcription factors, so that's good. So the T cells look more normal. In fact, none of them, they're all still dysfunctional. None of them secrete interferon gamma or TNF alpha, but if you take them out of, and treat them for 3 days with IL-15, then the T cells that had received the drug treatment are more able to be rescued than the control. Okay, so what we can conclude is the drug treatment is either decreasing or delaying fixed dysfunction, because you can still rescue more of them at the end. Okay? We wanted to show that this mirroring model also reflected epigenetic changes in patients, right? So we got tumor-infiltrating lymphocytes, PD-1 high, CD8 T cells from melanoma patients and non-small cell lung cancer patients, liver cancer patients do not get immunotherapy, so we're able to get these patients because they're enrolled in immunotherapy trials at Memorial. This is pre-treatment. Okay. And you can see if you do ATAC-seq in these patients versus naive and memory cells from healthy donors, the TILs from patients look different than the normal T cells. We wanted to compare to our mouse epigenetic signatures. So there's a problem that the patients and the mice have different genomes. So we use genomic liftover of non-promoter peaks from mouse to human, and the ones that can be lifted over, we look at accessibility changes relative to naive state in the corresponding species, okay? So, and then we just compare with a Spearman correlation, okay? So what we can say is, if we compare the TILs to naive, so the accessibility changes in TILs versus naive are most similar to this late stage dysfunction, to fixed dysfunction in the mouse model, okay? So suggesting that they're in this fixed state, that in the mouse model, this means they're resistant, they cannot be rescued by immunotherapeutic programming. In cases where the sites actually are orthologous, you can see some similar changes, like in this TCF7 locus, loss of event in the tumor, in the patients as well as the mouse. All right. If you want to have a biomarker that suggests that might be a biomarker for response to immunotherapy, and you believe that having T cells in this plastic dysfunctional state is better than in the fixed dysfunctional state, PD-1 is not a good biomarker, okay? So in the mouse model, all the T cells are expressing PD-1 from a very early stage, okay? But there are other markers that do distinguish between this early plastic dysfunction and fixed dysfunction. And we found, for example, CD38 and CD101, just by looking at gene expression changes, figuring out which ones were cell surface, encoded cell surface proteins, and then checking for antibodies, okay? And what we can say is that if you take, in the mouse model, T cells out at day 14, most of them are high for CD38 and CD101, these kind of fixed dysfunction markers, but some are still low. And if you take the ones that are low, you can, and treat them for 3 days with IL-15, so try to reprogram them therapeutically. The ones that were low for these markers can be reprogrammed, and the ones that were high can't be reprogrammed, okay? And that's the difference here in terms of interferon gamma secretion after treatment. Okay, so it seems like these markers, there are cell surface markers, at least in the mouse model, that distinguish between plastic and fixed dysfunction. All right, so this is, I think, the final result, and it's mainly for immunologists, right? So we know that an antigen in an inexperienced T cell, so a naive T cell put into the mouse, in the presence, goes into the tumor, recognizes presumably the neoantigen and undergoes some differentiation program that leads to fixed dysfunction, okay? So it doesn't achieve effector status or memory status, it becomes dysfunctional. What if you have an antigen-experienced T cell, so a memory T cell of the same antigen specificity? So this was done in the context now of established lesions. If you put these into the tumor, the short story, these are labeled in red, is that they also differentiate to fixed dysfunction, okay? No different from naive T cells, okay? And I think I'm running out of time, and I will just summarize and tell you really quickly, if I can, about a few other things going on in the lab. So just to summarize the study, and the paper is out as of a few weeks ago, we defined epigenetic states of tumor-specific T cells. We showed that they differentiate to dysfunction through two discrete states, this plastic state that can still be rescued therapeutically, and a fixed dysfunctional state that can no longer be rescued through immunotherapeutic programming. And these states coincided with chromatin states as defined by ATAC-seq, transcription factor binding analysis of the accessible atlas, identified transcription factors that could explain accessibility changes and were potential drivers of dysfunction. In vivo pharmacological modulation of transcription factors that were pulled out of this analysis was able to delay or decrease fixed dysfunction. Patient-derived PD-1 high CD8 T cells display an epigenetic signature of fixed dysfunction. And we found cell surface markers that can discriminate between these plastic and fixed states. We also showed that memory T cells with the same antigen specificity also differentiate in the tumor model to fixed dysfunction. Okay. We're doing a lot of projects like this with our very brilliant immunology collaborators at Memorial Sloan Kettering mostly. Many of these are published, some are coming out, and the next one to come out will be about the epigenetics of tolerance to self-antigen. So I think that's, it's a dysfunctional state related but not identical to tumor-specific dysfunction. What we're going to do next with this data set, so the data is published and you should take a look at it. It's very beautiful ATAC-seq data, but we haven't done the kind of modeling that we would like to do, and we're working on it now, trying to predict expression changes from the accessibility and DNA sequence content of ATAC-seq elements, and also build predictive models of the accessibility changes. We've published similar kinds of models in a different context. If you want to start understanding some of these changes in accessibility, I think you need to start thinking about 3D architecture of the genome. These accessibility changes aren't all independent, and we imagine that some of the enhancers are looping to promoters, and that's how they can affect gene expression. And we're working on statistical methods for understanding chromatin conformation capture data like Hi-C and next Hi-chip, which I think is feasible in our the mouse models we like to work on. And finally, another thing we're working on, if you want to understand gene regulation and the role of regulatory elements, you would like to delete them, edit the genome and see how they affect gene regulation. And in a collaboration with Andrea Ventura, we work to improve the tools for guide RNA design for CRISPR experiments, because the tools did not allow you to target the targetable genome. They were biased towards genes and also didn't take care of off-target effects very well. Okay, so this tool is out, GuideScan. Can use it. And one last thing is, I told you right at the start that we are interested in understanding gene expression programs in tumors and dysregulation of expression programs in tumors, and interpreting the role of somatic alterations in terms of regulatory programs. And one nice model was done by my postdoc Hatice Osman Bioglu, published earlier this year. And we're going on to look at ATAC-seq in cell line models for tumors and using both promoter and regulatory enhancer elements in this modeling of expression programs in tumors. Okay, and that's all I have. I want to thank Andreas Schietinger's lab, and all the analysis was done by an extremely talented PhD student in my lab named Lauren Fairchild. I'm happy to take your questions.